Yes, this is my micro. Okay. Okay. Thanks for coming. For me, it's a great pleasure to have here Bernhard Seifert, that I met first time in September 2013 in Innsbruck in a workshop of marine ecology. Well, he has been studying ants all his life. And he did a thesis about comparative study of the habitat selection of ants in central and southern part of the, at that time, German Democratic Republic.
Deutsche Demokratische Republic, behind the Iron Curtain.
But
this time,
as he's an extremely meticulous researcher, in 2017 he published a revised version of his all data from this thesis and a lot of more and communities, updating then all these names with the new names for taxonomy. And well, most of his scientific career has been developed at Senckenberg Museum of Natural History in Görlitz, Germany. And he's also the author of a very interesting monography about the ants of Central and North Europe, which contains not only taxonomic information, but a lot of information about ecology and biology of these European species. He's probably, but this is my opinion, the most important taxonomist in the world at the moment. But he's not only an taxonomist, he's also a field naturalist with a huge knowledge about birds and mammals, as he has been demonstrated during these days in Doñana when we were in the field. And now, well, I let him to speak.
Okay. Now, ladies and gentlemen, it is a pleasure for me to speak here in Sevilla.
Sevilla has been known for me only in two respects. That is the opera, the Barber of Sevilla, and the second one is Doñana National Park. So these two, and otherwise I have no knowledge about Sevilla, nothing. So yes, it is our topics today, wood ants. We have not so much talking speed time, so I will stop. You see here, redwood ants, the dominance of temperate and boreo-montane forests. This is, I will show you no overestimation. So, what's happened? Must check. What is here wrong? What's wrong here? I must go. Oh, please give it. Okay. So, this one. I have probably go to this mode. Oh, okay, it worked. But why doesn't it work? Oh yeah, now it works. Okay, yeah, yes. Between these images, you have 35 years. These images, when I started to write my first ant book in 1996 or 1994, we used the old system with films, where you got 36 films, and after 2 weeks you got aware that no image was good. So that was the time, and so we have lived in this time in a completely different world. The technology was, I say, Mesolithic or maybe Neolithic, and now we have the modern times. So, and this one is here from the Schwarzwald in Germany, and you see here, this is me taken with this old technology, and this is the newest one by Ingo Arndt, who provided a lot of photos. I helped him in his book on wood ants and also in his papers in National Geographic for knowledge, for the texts, and so on. So, yes, only that one, 1993, and this is 30 years later. So, okay, this is Ingo Arndt. He's a professional photographer who produces such pictures here, and it's nearly as we had get with a digital microscope, even better. So, he is here photographing ants, very well sophisticated. He took 1 year, it took him 1.5 years to take these photos. So that is only to give you the source of many of the photos. If there's no Ingo on, it was me, usually. So, the Formica rufa group. So, I made, among the taxonomic revisions I did, it was the one of the Formica rufa group in 21, I think. So, and I checked for the whole Palearctic 45 available names. Of these were 23 junior synonyms and 8 could not be interpreted. That means no types available. The description, the original description is useless, so they were deposited under incerte sedes, that means uncertain places, or basically. And the remaining is that we have 13 valid species, and it is likely that this number will not increase. As one of the few ant groups where we think, or where I think we are nearly at the end of the figure. But you see the many synonyms here. So, and these are the species. These only one, oh, it is shifted, I see. This one is East Palearctic, and this one is East Palearctic, and this one is East Palearctic. All the other species are in Europe, or found in Europe. Yes? Okay. That is quite a good thing to say. We will not have much surprise next. But the surprise lies in other fields with these ants. So, how I will only shortly explain how revealed how I got this taxonomy. Here you see the sampling sites, not the samples. The sampling sites from where I got wood ant samples. You see it goes to Kamchatka, and it's also in Iberia. Of course, many in the northern countries, and Central Asia, where I made several expeditions here to Kyrgyzstan, the Tian Shan Mountains, and so here's the Baikal region, and so on. These are samples, and that means I have some about 3,000 samples of wood ants investigated, and in each sample I took 5 workers, and sometimes I investigated 5 workers per sample. You can see that I had to generate by my numeric system some 300,000 primary data via the stereo microscope. These are measurements, countings, to give you an inspection. How broad is my noose at the base, how many bristles I do have here, and such things. How long is my scape, is it oval in section, and such things. So I took many numeric data. Without this, it would have been impossible to have this clear structure, a rather clear structure, but nature nature is responsible that the structure cannot be clear even which method you use. That is the disadvantage. So, okay, these are, and such a sampling cannot be done by geneticists. They have here, we have, it's the same done by genetics. We have only 12 localities from here to here where we got samples, or for 15 localities where we got samples. So that's another problem. So, and I want to give you only an example. This is not wood ants, but it doesn't matter. What does a taxonomist do? Because original descriptions are not reliable or often useless, you must get access to the type specimens. That is one. So, and you see here the age of the type specimens, the names, when it has been described. And then you have here 3 probabilities. For the species A, this is Formica exacta, this is species B, Formica foreli, and this is Formica brasilabris. So, and these are the museums where these types are kept. It is Helsinki, it is Geneva, it is Genoa, it is Brussels, it is Moscow, it is Lausanne, and it is St. Petersburg. And from these institutions, I got the types on loan and investigated them. And then you see, here's the clusters. This is the exacta cluster, this is the foreli cluster, and the brasilabris. And the red ones are the type specimens. So, and they are in this cluster analysis placed here. You see, here we can see, we have in the exacta clusters 5 names, and in these 5 names, the first available name is, of course, which is a name giving for this cluster. All others were synonyms. The same is with thoroughly, described in 1999 by Emery, and there are 3 taxa, 2 taxa described by Kutter and another one by Dlusky, the Russian. And Prasilabris is the same. We have only 2 described taxa. So Rufo maculata was later. So the name is Prasilabris. So this is a problem. I don't know. Genetics has difficulties to do this job because the type specimens are fragile, very old. And if you try to extract DNA from such type specimens, it is extremely difficult, and the curators, the responsible curators, will not allow it normally because the damage of the precious type specimen is likely. And on the other hand, the question if the DNA is still analyzable, or if you can. Now the situation is better because we can concatenate fragments, but it is a difficult thing. Normally, a geneticist cannot do, he can say it is a cluster A, B, C, but he cannot say what the name is. So, okay, and let's go into the more deep in the topic. You have here sampling points of Formica lugubris group here in Europe, and here you have northern Italy, Alto Adige. You see here a line crossing to Quebec in Canada. That means in 1971, 1.3 million workers in 2003 were transferred to Quebec. In 2005, the super colony has grown to 95 living mounds and 90 million workers. So it was really successful. But today, nobody would do that because it's strictly forbidden to introduce alien faunal elements into the area. So, but this was an argumentation owing to the absence in Canada of a suitable, aggressive, predaceous species of redwood ants and the conviction that an efficient, facultative predator of this type would be beneficial to the forest ecosystem. It was decided to introduce Formica lugubris from northern Italy. Today, strictly prohibited, of course, yes. So, and was it really Formica lugubris? It was always told this is Formica lugubris. So I did a cluster analysis to compare the next similar species. This is here the Formica paralugubris, which I described in 1996, and this is Formica lugubris. And you here in this system, this hierarchical system, you see 3 misplaced samples. Here is a sample misplaced, here is a sample misplaced. This is a total within, you see, about some 1.10, in this system 1.9% error, in another system nearly 0% error. So what is it? This is lugubris, and if you look at here, at this branch here, it is enlarged here. This is Canada, Quebec, and this is Italy, Peyo. This is these are the neighbors in this tree. So it's quite the locality of origin coincides coincides with the Quebec ants. So it is a tree based completely on morphology. So, and if you make a good job, you can even tell apart local populations by the system. In some cases, it is possible. You can say this is an ant from Kyrgyzia, this is an ant from north of Italy, and so on. Sometimes it's possible, not always. But if you have done a good system, you can do it. Okay, that you know, it is more in Spain. You will have it more in the north, in the Pyrenees. This is an aerial picture made by a drone of one large nest, and Formica polyctena is in this case the species. This is the species which makes the largest ant nest in the world, definitely, because we have here a nest with 95 square meters ground area, 16 million workers plus 20,000 queens, and with a territory of less 10 hectares, that's 20 football fields. And the largest nest, this is not the largest nest here. That is all the nest here. You see here the excavations of soil. not so large. The largest nest found in Belgium, in Germany, had a perimeter of 30 meters and 22 square meters. So the population and the fresh weight of the whole population is much larger than in the leafcutters and much larger than in the Dorilus wilberti, where we had 60 million workers, because the mean size of the Formica polyctena is larger than the mean size of a Dorilus. So this is also nests all over the world with the biggest worker mass inside. So Formica polyctena. So you see here, this is a German managed forest, commercial forest, only spruce, boring forest, and you see here nest by nest. So these commercial forests have been very strong populations, but due to the climate warming, spruce is dying on vast areas in Germany now, so that these ants got depressed by climate change, at least at the moment. So, and this one is also a commercial forest. You see very nice and big mounds. So, and this is the architecture of a wood ant. I will be quite short. The basis of a wood ant is often a tree stump. And you see here the excavations done by the ants in the tree stump. And the next one, better not to speak in detail, but this is a proof from National Geographic. I had to check. You see here the different regions of the nest where the larvae and the pupae are called, where the queen is, and then the whole yearly cycle of the ants. To make this paper, I had an email exchange with those people in Washington, which are the editors of this. I think I had 60 email exchanges with these, because they want to each detail know, and they say you have simplified, you cannot avoid to make an error. If you simplify, that's unavoidable. So we had to think, but in the end, they got it. So what is the mating with the ants? The matings, these males are too large to mate in the air. In Formica, the mating is always on a solid surface in any Formica, except in any Formica. So that is a mating. And of course, the queens have a low success rate. There are estimates that 1 in 1,000 or 1 in 2,000 queen can found a colony if it tries to do it in temporary social parasitic way. That means the queen tries to enter a nest of a slave ant species of Cerviformica here, to kill the queen, to get rid of the queen, and to use these worker ants to start the colony. After this, it becomes independent. So they can live on their own, they can do anything, they have to do any work what is necessary. So it's only for colony foundation. So these temporary, these ant queens, these types of ant queens which try to colonize or to found as a single ant, have a lot of enemies and they are frequently killed by the attacked host ants. In this case, it is Cineraria, which is much too aggressive. The success rate is nearly 0 in Cineraria. In Cunicola and Fusca, it's better. So, okay, and you know, these are different systems. Yes, this is a Formica rufa polyctena case. You have here the typical monogenous Formica rufa. It has 240 ovarioles and lays 300 eggs per day and so on. That means so for per season and 1 million for the whole life. And it can live at least 25 years. So this is a monogenous case. It's very different. But the polygynous is in this case polyctena, but rufa also expresses a polygynous societies where with many queens. So we can take it also for these data also for the polygynous societies of Formica rufa. There's a very different situation, 10 eggs per day and per season, and maybe 7,000 for the whole life, and the longevity is maybe around 5 years. So it's not very long. You see here the different stages. This is the egg. Normally, the vernacular saying is that these are ant eggs. In Germany is what is called the pupae, and these are the larvae. And you have here small larvae in this picture. So you have two different types of ants, which may occur, as I said, within a species, within forma garufa, and these two types, these two social types, are directed by supergenes. So it's only an impression. Sometimes they have naked pupae. The normal thing is that we have a cocoon. So you see here, when the ants hatch, there's help. Ants also do help each other in immune defense and such things, and of course in cleaning, and if some ant is under soil and needs to be excavated and so on. So mutual help is something. So now we come to the first astonishing thing. Because anything what I said is are old hats. So this is a nest, a large nest covered by more than half a meter of snow. And we know such situations are normal in Finland, they are normal in Russia, they have been normal in Germany before, now no longer normal. So, and these nests in Finland, we have investigations in Finland and in Russia near Moscow, that the development began under the snow cover at low temperatures, independent of the weather situation outside. So they can activate, they are under this snow cover, but independent from the current weather situation. They do it if there's a lot of snow, that is normal, but they do it also if there's no snow or little snow. So, and this is one of the excavation made by Gennady Gluski near Moscow. You see here, this blue one is the air temperature. This mound was covered by half a meter of snow near Moscow, and it's April. And there's no this is the inside nest temperature. There's basically no change. And then suddenly, the nest core was heated by 21 Kelvin or 21 Celsius within 33 hours, steeply heating up, 20th April to 21st April. And this heating proceeds independent of the weather situation. So, and this is possible only for populous nests which have young workers, which have stored in autumn of the last year enough fat. So the heat, and it's very clear that it's a heating via muscular activity similar to the honeybees. So the question is now, okay, that is a fact. But how can the ants determine the right point when to start up heating? Basically, such a wood ant nest is like a mammal which during hibernation, a squirrel or a hedgehog, they should have low temperature during hibernations and avoid heating up because of the loss of energy, not at the right time. If a hedgehog heats in Germany in January, he would die. So it's better to heat when the spring comes. And what is the most reliable indicator for this? This is day length, and it is the day length which is reflected by the orientation of magnetic lines. That is the reason. So another example here, this is from Finland, basically it's the same, done by Rosengren in 1987. And this is from Czechia. You see here the mean nest temperatures, the thick and the environmental air temperature. You have here one nest only amongst these nests from Czechia which could do this rapid heating. All others depend on sunning and other forms of activation. So, okay, and I said it has not been proved, but day length is clear. That is the right timer for heating up. That's the best timer because it is independent from the weather. But probably the ants can realize the magnetic lines, which depends on the height, on the azimut of the sun. That is my theory, and at least we don't know if below a half a meter of snow maybe there is some light they can recognize some light. But it is clear they can recognize it independent from what is going outside. So it must be an astronomical cue for this heating up. And there's no other solution. So, okay, you see here the spring bathing of those nests which have not the possibility to heat up independently. Spring bathing here in the center is the queen. This should be maybe Formica rufa. So, or you see such clusters here on the mound surface, and you see here a false color thermal image. So that is one thing. So, and how do the wood ants feed? I think I shortened this a little bit because there's nothing very new. They make direct use of vegetarian food, phytophagy, nectar, pollen, and they cut sprouts. That is basically direct use of phloem sap, and they use the elaiosomes of several plants. You could speak about 1.5 hour about this system. This is widely spread in temperate forests where ants are responsible for the distribution of seeds. They only use these elaiosomes. The seed itself is of no interest, but they distribute the seeds because it's interesting, and the seeds are very similar in the composition to some larvae they have. So then is another thing, preying on live animals. Here are different examples, and you have your carabid beetles. I have checked it with large carabid beetles and put it directly, immediately to the ant trail, and they attacked it. But the large carabids, Coriaceus, for instance, they went away, and so no, they cannot catch it. But the problem is another. The beetle got many shots of formic acid, and this formic acid will disable the beetle somewhere in the bushes, and they take it when he's dead and put it back. But the direct experiment seemed they cannot catch a carabid beetle. So that is one thing, and this is another one, the beautiful Carabus intricatus. His beauty did not help him to become saved. So, and this is very rare, that the pillage of beehives. Here you have the formica ants in a beehive. They consume the larvae and the pollen, and of course the honey and so on. So quite, but it's very rare. That's not so rare. So, and how strong is an ant? We have a lot of things. Here, this is a photo of mine. This was an astonishing thing. There was a thick tree cut in 10 meters height, and this cut as it broke, and this was a plane of such a size here, and they erected their mount on this tree in 10 meters height, on the plane, and the cut plane, and they needed to take up all the materials laboriously up the tree. So, and the stick here weighs about 200 milligram, and the ant is about 10 milligram living weight. Yes, so they are very strong. The ants, you know it, and this is also feeding on animals. This is so funny. 3 hours later, you will not see this mouse. Something has left from this mouse. Yes. Okay. And then we have trophobios with plant sapsuckers, or that I call disguised phytophagy, because the aphids it is easier for the aphids to have a permanent flux of phloem saft through their body and to give it to the ants. This is sufficient for them to exploit enough of the amino acids for their own growth. So it is quite near to phloem saft. So it is, the trophobios is basically disguised phytophagy. Now it's interesting to make an economic point balance, or in German, betriebswirtschaftliche Bilanz. So, and this is a model. The coverage of energy needs is about 80% by trophobiosis because there are much sugar in, and 18% by, and only 2% in the end by phytophagy. So it's quite meaningless, but the anabolic requirements for making own body protein, this is basically the oil to run the system, and this is basically the system to build up body tissues. So, and this is the main contribution is done by prey, and 32 by trophy, and only less than 1% by phytophagy. And what is the efficiency of an ant nest? Ant nests are high security systems. It is difficult to destroy an ant nest. Once it has been founded, the eradication is difficult, and we have an efficiency, production biology. Only 4% of the energy retrieved to the nest is transformed into ant biomass. That means 96% are lost, largely by metabolism for foraging and defense, colony construction, and also they have an incomplete utilization of food. Many food particles are wasted, not entirely exploited. Then the maintenance of a million army is cost efficient, and not cost efficient, is costly. And so now I have another model calculation for a woodland system at saturation density. That means this is the maximum capacity a woodland may hold for wood ants. And here we have a population of about 90 kilograms fresh weight, or 8 million workers. And these would consume 4% of the net production of this forest, net primary production, but only transform 0.16% of net primary production into ants. That's very little. But if you walk in such forest, you cannot go, because with every step you kill some 20 ants. So it's also noisy, these forests, because always you hear the footsteps like rain. So it's immense, but it is only 0.6% and harvested only 4% of the net primary. It's quite few, even if they are apparently the dominant thing in this forest. So any enemies and enemy defense, yeah, you know, the poison bladder of a formica, 2 milligrams, that is much. If this is perhaps for major workers which have 12 or 13 milligrams fresh weight, so it's 1/6 of it is acid, formic acid of 65%, and formic acid is is water soluble but also lipophilic, so it can penetrate the cuticular and go into the ant. So the ant prey which has been sprayed will not die suddenly, but later. And this is one of the most efficient weapons they have. So you see here. So, and this is, you see, as a New Eve's fireworks, but without noise and dust. So what they make here, and this is quite nice. And this is what we see. This is from northern Germany near the Baltic Sea. You see here a wood ant mound heavily damaged by woodpeckers. This is a winter situation. What is very little known, that bears eat much ants, in particular wood ants. You know, boreo-montane forests, there's a high wood ant density, and you have it here. We have investigations from Sweden over Central Asia to Japan, where the Yezo brown bear is the same species as in Europe, and Kamchatka is the same thing. So we have 30% of the energetic part of the food of this guy a year. This is proven from Sweden east to Japan. And now I made some estimation by the data given by the Russians on the bear density. Wow, we have about brown bear density about 1.5 individuals per 10 kilometers. That means 3 by 3 kilometers, 1.5 bears. quite a lot. So, and the capacity limit is here 20,000 kilogram fresh weight on the same area, and these bears would consume 3,600 ants per year, a kilogram, and this is 18% of the whole wood ant biomass the bears consume. And what is likely? They have an extremely long tongue, the bears. Now, not like the ant bear, but it's long. And I have seen in Central Asia, walked in a canyon, a steep canyon with a very steep rock, very steep rock, and we found such blocks here removed by bears. And you have, if you remove such a block, you have a plane surface, and you can make it and have take up enough ants. But I went back because there was no escape for the bear in this situation, so I didn't go further, and went back up to my comrades. And what was interesting to see, these huge blocks which they removed and cleared of ants. So, okay, this is one, you know, a picture series of the ant lion in action. Here one, he has gripped his as a two of the as a leg of the, and here is a drawn into the soil. So, and another story is the infection, and you have here so-called, you have here so-called ascomycetes. In this case, it's agaricella, which is sometimes extremely abundant on the ants. This is a Formica, I don't know, maybe a Formica polyctena. It is not, it's rarely found in monogenous ants, but frequently found in polygynous and polydomous ants. Ants which have hundreds of queen, have hundreds of interconnected nests. So it's clear, if nests are interconnected, fungus may spread. They may infest each other, so that these polygynous society are more heavily infested by these fungi than a monogenous society, where they can clean up and where the clean is cared for by 100,000 or 80,000 or 50,000 workers. So it is another situation. So, and this is one, you see here such a detailed photograph of such an ant. Here's the Formica rufa. They don't cause damage, these fungi, to the ants. They can do anything what they want. Sometimes the joints are blocked when the fungus is here, so they cannot move. But it has no effect on the interior of the ant, and also no effect on the activity. So, and this is what we call the summit disease. Writing error. It doesn't mean summit disease. That means the ants move on top of grasses or other plants and bite into the grass and die. And this is done by what is Dicrocerium lanceolata, I don't know now the English description. It is a trematode. But it is also by Pandora myrmecophaga fungus. You see the same appearance but different sources. Here you see the fungus, and this is another one. So, and you can also die during standing if you have infection. This is a Camponotus ligniperda major. I got this picture by Michał Kukla, Polish one. So, apropos fungal infection, it is well studied now that the chemicals inside resin help to prevent infections. And so, wood ants regularly take up particles of resin, move it onto the mounds, so that fungal growth is limited, and also other infections are limited. So, on this one, myrmecophiles is not to begin talking about these. There's only one example. This is here the Clytra quadripunctada, is a chrysomelid beetle. Here is depositing eggs. It looks like pineapples, like a corn, like cones of conifers. It is falling down onto the ant mound, wood ant mound, and here these beetle larvae construct hulls of their feces, and these hulls are very hard, like stone. And you see here the ants are walking on the mound, and here, when the ants are not here, they look out and consume something in the nest. And when the beetle ecloses from the pupae, he has a problem because he's then attacked. So the larvae are wise if they move to the periphery of the mound, as that the beetle has not too long walk inside the nest, so we can leave it quite easily. So it is the most dangerous. The other one is they are completely safe. Nothing is done, no harm is done. And this is another problem. I had some lecture alone on this anting. We have passive and active anting. This is the passive anting. In passive anting, it's quite clear that is a defense against infestations, against infestations. So, and hybridization is another problem. This is in wood ants a venue of adapting to rapidly changing world. You know, we have habitat destruction, fragmentation of woodland, we have a climate increase of climate. So, and we only to show you this is a simplified diagram of what happens in the Central European wood ant. You see here nearly each species hybridizes with others. These are the region in which the hybridization occurs. So, and one species is supposed to be even a hybrid or genus species, like the Italian sparrow, for instance. So, and that is the situation basically. And we have two types, main types: the selection, the Formica rufa and the Formica polyctena. So, and these are selection stabilized with ant species. So they have different morphology, colony demography, dispersal strategy, temporal and spatial pattern of swarming. So, and this is the first case, is hybridization as adaptation to the fragmentation of habitats here, the fragmentation of woodland. So here you see, Görlitz is about here. Here you see an aerial picture of the landscape. Here in the north you have large forests, coherent areas, large forests, and here, this arable land, you have many small woodland patches, 1 hectare, 3 hectare, 2,000 square meters, and so on. Many. And in this encircled area, 27% of 212 nests belong to hybrids between rufa and polyctena. In this area here, where you have a compact woodland, you find much lower frequency of hybrids. So, and you see here what I found when I came to Görlitz, when I studied these wood ants. You see here the morphology. This is here a pilosity index. You have here in the compact system a huge peak of intermediate, in the nests of intermediate samples, and in the compact woodland system, the thin line, you have only few hybrids. So that is the situation. And the hybrids here, they combine the dispersal, reproduction, and space utilization strategies of the parental species. And they have selective advantage in fragmented woodland structure. So they combine, I will show later, the potency for local dominance by making polydomous colonies with the ability to make a far-ranging dispersal flight. They combine two things: fly over 1 kilometer or 2 kilometers of arable land to the next forest, finds there a Cerviformica species, found a single queen independently in a socially parasitic way, and then grow up in a nest and then split, make a splitting and dominate these small spots. So that is a combination of two things. The one strategy, fly away, a long-range flight, is a typical Formica rufa strategy, and making polygynous colonies with huge numbers of workers and queens and many nests is a typical polyctena strategy. And the hybrid does better in these fragmented systems. So you show here these black spots are the Cerviformica, nests to found for socially parasitic colony foundation. Here you have fragmented systems. The X are the hybrids, the R are the rufa, and the P are the polyptena. So you see here the Formica rufa is a typical long-range dispersal. It's good in social parasitic colony foundation. It is a queen dominant species where the rufa have a odor dominance, and the supercoloniality is biggest here in the Formica polyptena. So, and this is what we found. You can see that the hybrids versus Formica rufa is clearly distributed different, and also the hybrid versus the other parental species is clearly different in these medium-sized fragmented and the large woodland systems. You see it's not quite a high number of woodland mounts I have investigated. So you see, and this is a model, a mathematic model. The parameters in this model are these here, not speak about it. We have a lot of parameters, at least 15 biological traits were simulated for 10,000 nest territories within an imagined plot. So you have here a ratio of margin field, meaning margin line fields is low. And in this system, the computer simulation predicts a low number of hybrids. And in the fragmented system of the same total area, you see here that the system predicts a higher number of hybrids. So it means that the observations and theoretical predictions are do match each other or are quite coincident. So alone this topic is a talk of 1.5 hours. So hybridization, next one, to rapid climate warming is another problem. So you know a mutation is very slow. Mutations can only work as quick adaptation if we have an extremely small generation time. Bacteria, of course, can adapt quickly. Viruses can adapt quickly, and so on. But a normal multicellular organism needs to wait once it gets a successful mutation. It is a long time, and the climate warming at the moment is 3 times faster. What we observe, the anthropogeneous climate warming at the moment is 3 times faster than during the change from the cold period in the Pleistocene to a warm period of the Pleistocene. So that is a thing you cannot manage by mutations, physiological mutations. It's basically impossible. So, and this is what we have is only a climate diagram. You see here the situation with the CO2, which has gone, you see here the separation with the temperature. And this one, what we have, this is a Mauna Loa in Hawaii observatory. We have 4,125% ppm of CO2 and methane in 2024. And the speed of temperature increase has been 0.1 degree 100 years, or 1 degree just a moment, 1.1 degree whereas 100 years, or 1 degree in 1,000 years. So it means normally the shifting from the cold period to the warm period takes some 10,000 years. That means some 10,000 degrees more. So that means we have now globally 1.3 in 46 years, or this is 2.8 in 100 years. So it means 20 times more faster than the natural increase. So it's a huge problem, of course. And this is the situation in the ocean all over, and some nearly 70% of the whole globe are covered by oceans, and the oceans are slow in reaction. So if you go to the continent, if you go to Görlitz from 1980 to 25, we have now 2.4 degrees in temperature increase in Görlitz. The more north you go, the more difficult the situation becomes. So, okay, and what would happen without man? The probably development without Homo exhalens is that we would have go into the next cold time, would have gone into the next cold time, but very slowly. You see here, this takes very, very slow. Much slower. You're seeing here, the warming is quite fast. The cooling is slow. The warming is fast. The cooling is slow. The warming is fast. The cooling is slow. I don't know explain why it is so, but it is a fact. So, what can an organism do to respond on climate warming. He can do a physiological adaptation, I said, by mutation impossible. He can await to a cooler habitat at the same locality. It means an ant living in a xerothermous crescent can go into the bushes or into the woodland to live there. No problems really for ants because they are omnivorous. They are not specialized to certain foodstuff. A phytophage, which is specialized on a certain plant species, cannot freely move into the forest because you will not find its food plants. But the ant could do it, and we can also observe it that Larsis amarginatus lives in Bulgaria inside shady forests, but in Germany it is at the hottest stone walls, in the hottest habitats. So, but the mean temperatures of both habitats are similar. So that is one. The possibility, or he can emigrate to a cooler geographic region. This is what happens. If the insects can fly, it is easy to go to cooler geographic regions. In Germany, we have so many new species due to climate warming that the taxonomists become difficult to say what it is. So, or it got extinct. And you know, if evasion to a cooler geographic region is not possible for animals living in the Yellowstone National Park because there's cultural land, streets over streets, and highways over highways. So they must stay there, die or survive. That is one. So, okay, what is in with wood ants? I said we have extremely high frequency of hybridizations, and this is what the Finnish, what we did together with the Finnish team, mainly a Finnish team. We have here the distribution area of the Formica aquilonia. It's in the Alps, higher up in the mountains. This is the area where Formica polyctena is. There is some mistake in this map, but basically it's true. Here is the main distribution of Formica polyctena, and this is aquilonia. So it's more northern or higher. The one is boreo-montane, and the other one is a temperate forest species. But in southern Finland here, and these sites here, they meet. And in this site, we have locally up to 70% hybrid species, and some regions, even if there's an isolation, you have on a semi-island 100% of hybrids. And these hybrids work, and the genetics in this hybrid population work like in a cooking pot. So very new variants are formed, and so on. So, and what the experiments were, we have here difference in thermal tolerance of the hybrids between aquilonia and rufa and polyctena. So we have here first so-called heat knockdown experiment. You see here in heat knockdown, as expected, the Formica aquilonia was the first to be knocked down, and then we have the hybrids, and the Formica polyctena was no difference here. So that means the hybrids inherited the heat resilience of Formica polyctena. So, and the opposite test on chill coma experiment, who could establish from chill coma, recover from chill coma, the opposite is the case. Here, of course, the boreo-montane species made best, and the others were slower. So we have here, by mutation, an immediate response to a changed environment. And so the hybrids have a better chance to get along with the climate change. And I'm personally convinced that wood ants are one of the least ants which will die out due to human activity. The knockdown now of the Formica polyctena in the German commercial forests is only an ephemeral phenomenon. They will recover. They will go into the deciduous woodland and will have stable populations. So, but the ants we have now and the wood ants we have in 100 years are not the same. That is the consequence of it. So, okay. Oh, I think it's quite good. I think it is okay. So I can stop my talk. Thank you for your attention.
Thank you very much. Good. Thank you very much for this talk. So we're opening it now for questions, but because we're the first time in this very beautiful but also very large room, we kindly ask you to speak loudly. So
yeah, yes, I'm completely deaf on my right ear, so you must, and try not to speak Spanish English or French English. Try to speak as a news speakers in BBC.
Otherwise, I'm here to translate.
Okay.
So, any questions in the audience?
Yeah.
I don't know if I like the microphone. Okay. The thing is, like, you have been talking about the hybrids, and we have seen the rufa hybridizing with different species. Mm-hmm. The point is, like, in many forests in which I have been working, I found Formica truncorum very close to Formica rufa nest, or relatively close. Can we expect future hybridization of these two species?
No, I think the species have nothing to do with each other. But it's, what do you mean? You compare the two species, or what do you want to know? No, that is accidental. This is quite accidental. The Formica truncorum is another type of wood ant. A little bit, it's split off from the tree of the Formica rufa quite early, and it's another guy. It is a guy who distributes very far. It falls from heaven. Now, if you have a small spot of, say, the size of this room, where is some rock with columnar and vaccinium, you went to the spot over many years and didn't see any truncorum. Suddenly it's here, and after 10 years it will disappear. So it is a very good in distribution, but also it has a chance to split up in colonies. You know, and the Finnish guys have had super colonies on the Scary Islands. So the complete scaries were covered by these ants. So it is in biology similar to this other ant, but it is more flexible and more distributing. If you may have it inside a city where you have never seen it, in a wall of a church, behind is a churchyard with some conifers. So well, in woodland, it depends on the situation. In Europe, these ants normally don't have huge nests. But in the Tian Shan, where we have no competing other wood ants, Tian Shan mountains in Central Asia, Tian Shan, we have huge mounds of these species which are attacked by the bears. We have seen the bears in these truncorum nests. But basically, if you found them together, it's accidental. Nothing to. Yeah.
Yeah, thank you for your talk. Yeah, my question is regarding the thermotolerance. You have found, or you present in the last slide, that there seems to be an adoption of the heat tolerance of the heat tolerant species, parental species. But my first question is regarding whether there is some sort of hybrid vigor in relation if whether the hybrid has greater thermotolerance than the parental species with heat tolerance.
A hybrid vigor is no rare observation in ants, in the worker ants. In some ants, the hybrids are as big as the larger species of the both parental species. So should we shut some of these systems here? Maybe we go only on to this microphone. So now, okay, there is a hybrid vigor, and hybridization is in the Stonolasus. I have got the impression that these hybrids are much more popular and stronger in the workers. But this is another type of hybridization. This is strategic hybridization. To use only the sperm of another species to found a nest, and then to produce workers. So that is this is an established system, And what is the advantage for such an ant queen? If she got not sperm of her own species, she can reduce the damage. Instead of having no progeny, I choose another species. So, and then the ant queen can 100% can give away or give further 100% of their genes via her sons, because the sons are completely the genotype of the mother, because ants are haploid. Yeah? So that is what I call social kleptogamy, others call it sperm parasitism, others call it asymmetric social hybridization. Yeah, that's what. So, and this is you have so many different types of hybridization within ants, so many awkward genetic mechanisms, as though it is a story for 3 or 4 evenings. So this is it's incredible, and you can even one and the same giant may even produce her own genes, but conserve in her genome the genes of another species, which has been recently found. So it's the most astonishing thing why we have.
Any more questions? Okay, if this is not the case, do we have anything online? No, okay.
Can I see her?
All right, thank you very much again for this talk, and see you all again next week.
Mm-hmm.